Good morning, dear students. Hope you are following my videos. And dear students, I will assure you 100% NCERT coverage in my videos, and wherever an extra information required to succeed in NEET and other CET exams. And in my previous video, we have completed with hemophilia and color blindness, and in today's video let us complete with the remaining 3 Mendelian or gene disorders. And now let me start with phenylketonuria. So phenylketonuria is a gene disorder or Mendelian disorder. The gene responsible for causing phenylketonuria is present on 12th pair of chromosomes. So phenylketonuria, it is an example for autosomal recessive trait, and you know recessive traits are always expressed in homozygous condition. Means when a person suffers from phenylketonuria, whenever a child inherits 2 recessive alleles from the parents for phenylketonuria, then only that child is going to suffer from this condition called phenylketonuria according to genetics. And phenylketonuria is the problem with the metabolism of an essential amino acid, that is phenylalanine. So important point, phenylketonuria is the problem always associated with the metabolism of an essential amino acid, that is phenylalanine. And students, remember, whenever we consume phenylalanine in the form of food, to metabolize phenylalanine by our body tissues within the body, we need an enzyme called phenylalanine hydroxylase. So phenylalanine hydroxylase is the enzyme responsible for the metabolism of phenylalanine within our body cells. And phenylalanine hydroxylase is essential for the conversion of phenylalanine into tyrosine. Means students should remember, phenylalanine gets converted into tyrosine in presence of an enzyme called phenylalanine hydroxylase. Then this tyrosine is utilized by our body cells and tissues. So always remember, phenylketonuria is caused due to the deficiency of an enzyme. Which enzyme? Caused due to the deficiency of phenylalanine hydroxylase enzyme deficiency. So whenever a child suffers from deficiency of this enzyme called phenylalanine hydroxylase, phenylalanine cannot be converted into tyrosine. As it is cannot be converted into tyrosine, it is stored in the body in the form of phenylpyruvic acid derivatives. And these phenylpyruvic acid derivatives, when they are accumulated in the body tissues, it causes mental retardation. And the same phenylpyruvic acid derivatives in the form of ketones excreted in the urine, and these ketones are excreted in the urine due to the abnormal metabolism of phenylalanine. That's why this condition is called phenylketonuria. So I repeat, students should remember, phenylketonuria is an example for autosomal recessive trait. Gene is present on 12th pair of chromosomes, and it is the problem associated with the metabolism of which amino acid? Phenylalanine. And phenylalanine is always converted into tyrosine in presence of which enzyme? Phenylalanine hydroxylase enzyme. So students always remember, in CET and NEET repeatedly asked question, the Mendelian disorder caused due to hydroxylase enzyme deficiency, that is nothing but phenylketonuria. The next and the most important gene disorder, that is sickle cell anemia. And students, The term anemia usually represents the presence of less hemoglobin in the blood. If you remember, usually in healthy adult humans, 12 to 16 grams of hemoglobin is present per 100 ml of blood. Whenever hemoglobin content decreases or quality of the hemoglobin decreases, oxygen carrying capacity of the hemoglobin also decreases, and whenever body tissues receive less amount of oxygen than normal, that condition is usually called anemia. And this anemia is caused due to various reasons, and today we discuss sickle cell anemia. And once again remember, sickle cell anemia is an autosomal recessive trait. Gene for sickle cell anemia is always present on 11th pair of chromosomes, and it is a qualitative problem. It is a qualitative problem, means in the condition of sickle cell anemia, the number of hemoglobin molecules synthesized is going to be normal, but the quality of the hemoglobin decreases, means the oxygen carrying capacity of the hemoglobin decreases. Hence, there is no change in the number of hemoglobin molecules synthesized in the blood, but the amount of oxygen carried by the blood to the body tissues decreases. Hence, it is a qualitative problem. And coming to the most important aspect of sickle cell anemia, why this is called sickle cell anemia, I have represented with the simplest diagram. You know that in adult mammals, RBC are biconcave and disc-like and enucleated, without nucleus. This biconcave shape offers more surface to volume ratio, so that biconcave shaped RBC can carry more oxygen to the body tissues. But in this condition, biconcave disc-like RBC are converted into sickle-shaped RBC, or they are converted into crescentic RBC. So, when RBC gets converted from biconcave disc shape to sickle shape, oxygen carrying capacity of the RBC decreases and person develops symptoms of anemia. So what is the important reason for anemia here? By the formation of sickle shaped RBC is responsible for anemia. That's why this condition is called sickle cell anemia. And coming to the technicality of the sickle cell anemia, why biconcave RBC gets converted into sickle shaped RBC. And here the problem is always in beta chain of the hemoglobin molecule. And students know hemoglobin usually contains 4 polypeptide chains. In these 4 polypeptide chains, 2 are the alpha chains and 2 are the beta chains, and each alpha chain is made up of 141 amino acids and each beta chain is made up of 146 amino acids. And students, remember, in sickle cell anemia condition, where is the problem? Problem always lies only in the beta chain of the hemoglobin molecule. And what is the problem in the beta chain? I have written here, in the beta chain, at the 6th position, amino acid glutamine is replaced by valine. Means, in healthy hemoglobin in the beta chain, at the 6th position, which amino acid should be present? Glutamine should be present. But in sickle cell anemia, in the beta chain at the 6th position, glutamine is replaced by valine due to a gene mutation. When glutamine is replaced by valine, structure of hemoglobin molecule gets changed. And if you remember, in chemistry you will study what is the primary structure of a protein? Sequence of amino acids in its polypeptide chain. Here it is very clear. Sequence of amino acids in the beta chain of the hemoglobin molecule is changed. So with this it is very clear. Hemoglobin primary structure is changed. And that hemoglobin structure only is responsible for biconcave shape of the RBC. As hemoglobin structure is changed, RBC shape also gets changed from biconcave to sickle shaped, and person develops symptoms of anemia. That's why this condition is called sickle cell anemia. It is also an example for autosomal recessive trait. Problem is always in beta chain of the hemoglobin molecule. What is the problem? In the beta chain at the 6th position, amino acid glutamine is replaced by valine. In sickle cell anemia, glutamic acid is replaced by valine in the beta chain of the hemoglobin molecule at the 6th position. What is the reason for the replacement of glutamine by valine at the 6th position? It is just because of a gene mutation or a point mutation. What is that mutation? Simple terms, if the question is asked, the mutation responsible for sickle cell anemia, the codon GAG is replaced by GUG on what? mRNA. Students, you know, you have already studied. Codon is a triplet sequence of bases. Codons are always present on mRNA, that is messenger RNA. A particular codon codes for a particular amino acid. So here it is very clear. On mRNA at the 6th position, if GAG is present, it codes for which amino acid? Glutamine. But GAG is replaced by what I said? GUG on mRNA. That's why instead of glutamine, GUG is coding for valine. That's why when on mRNA, GAG is replaced by GUG. So in the beta chain of the hemoglobin, what happened? Glutamine is replaced by valine, and which caused the change in the shape of RBC from biconcave to sickle shaped. So it is very clear, change is happening on mRNA, I said. Why this change in mRNA? Can we expect mutation to directly occur on mRNA? No. Mutation actually occurs on DNA. So what is this mutation on DNA? See, I have written on mRNA, codon GAG is replaced by GUG. Adenine nucleotide is replaced by uracil nucleotide. Why it is happened? If you ask me this question, a gene mutation or a point mutation was taken on DNA. Means on DNA what was the sequence? CTC was the sequence. When on DNA sequence is CTC, on mRNA it is going to be coded as GAG. But on DNA what happened? Due to a point mutation, thymine nucleotide is replaced by adenine nucleotide. Means on DNA only CTC is replaced by CAC. That's why on mRNA what happened? GAG codon is replaced by GUG. So if you ask me what type of gene mutation it is, I say it is transversion. Once again an important question which is not directly mentioned in the textbook. If the question is asked, which type of gene mutation or point mutation responsible for sickle cell anemia, our answer should be transversion. Why I am telling like this? You know, the two important types of point mutations are transition and transversion. In transition, what happens? One purine may be replaced by another purine, or one pyrimidine is replaced by another pyrimidine. But in transversion, what happens? Tell me. Purine is replaced by pyrimidine or pyrimidine is replaced by purine. You can see here GAG is replaced by GUG, I said. Adenine is a purine, you know already, and it is replaced by uracil. Means uracil is a pyrimidine. So what happened here? Purine is replaced by pyrimidine on mRNA. You can look on DNA also. CTC is replaced by CAC. Thiamine is a pyrimidine. Adenine is a purine. So pyrimidine is replaced by purine on DNA. Purine is replaced by pyrimidine on mRNA. So you can consider both. Ultimately what happened? Purine is replaced by pyrimidine or pyrimidine is replaced by purine. So which type of mutation it is? Transversion. So the type of gene mutation responsible for sickle cell anemia is transversion, and the change on mRNA GAG is replaced by GUG, and the change on DNA CTC is replaced by CAC. And finally, remember, sickle cell anemia is a autosomal recessive trait. Once again, when a child suffers from sickle cell anemia, whenever a child receives two recessive alleles from the parent, then only child suffers and shows symptoms of sickle cell anemia. But it is a rare case because recessive traits are always expressed in homozygous condition. So students, remember, most of the people in human population are heterozygous for sickle cell anemia trait. Means they have one copy of recessive allele and another copy of dominant allele. So that's why they do not show symptoms of sickle cell anemia, but such people can pass recessive allele to the next generation. And also one additional information you have to remember, people who are heterozygous for sickle cell anemia trait shows or exhibit a high degree of resistance to malaria. So finally remember, sickle cell anemia is an autosomal recessive trait. Gene for sickle cell anemia is present on 11th pair of chromosome. What is the major problem in sickle cell anemia? Biconcave RBC converted into sickle shaped RBC. And why this change? This change is because change in the structure of hemoglobin molecule. What is the change in the structure of hemoglobin molecule? Change happens only in which chain of the hemoglobin? Beta chain of the hemoglobin. And what is the problem in the beta chain? At the 6th position of the beta chain, amino acid glutamine is replaced by valine, which causes the change in structure of hemoglobin, which ultimately changes the structure of RBC from biconcave to sickle shaped, whose oxygen carrying capacity decreases greatly, and person develops symptoms of anemia. Person suffers symptoms of anemia due to the formation of sickle shaped RBC. Hence this condition is called sickle cell anemia and it is a qualitative problem. Coming up with the last Mendelian disorder given in the syllabus, that is thalassemia. And once again, thalassemia is also an example for autosomal recessive trait. And students should remember, in thalassemia also person develops symptoms of anemia. As we have discussed in sickle cell anemia, in thalassemia trait also person develops symptoms of anemia. But if you ask me, what are the main differences between sickle cell anemia and thalassemia? First difference is that sickle cell anemia is a qualitative problem. Means there is no decrease in the number of globin molecules produced there. But thalassemia is a quantitative problem. It is the problem associated with the synthesis of very few globin molecules. You know, hemoglobin. Hemoglobin is a conjugated protein. It includes heme plus globin. So globin is the protein portion of the hemoglobin molecule and heme is the non-prosthetic group. So non-protein portion, and we call it prosthetic group, right? We know that. So globin chains are nothing but the protein portion of the hemoglobin molecule, and I already told you. In each hemoglobin molecule, how many polypeptide chains are present? 4 polypeptide chains are present. That is 2 alpha chains and 2 beta chains. And one more important difference in between sickle cell anemia and thalassemia. In sickle cell anemia, problem is always in which chain? I said beta chain. But in thalassemia condition, problem can be in alpha chain of the hemoglobin molecule, or the problem may be in the beta chain of the hemoglobin molecule. And if the problem is in alpha chain of the hemoglobin, we are calling it as what? Alpha thalassemia. And if the problem is in beta chain of the hemoglobin, it is called beta thalassemia. So students should remember the two important differences between sickle cell anemia and thalassemia are: sickle cell anemia is a qualitative problem, whereas thalassemia is a quantitative problem. And in sickle cell anemia, problem lies only in the beta chain of the hemoglobin molecule. But in thalassemia, problem may be in the alpha chain of hemoglobin or beta chain of hemoglobin. And when we discuss alpha thalassemia is controlled by 2 genes. And alpha thalassemia is always controlled by 2 genes. And these 2 genes are named as HBA1 and HBA2. And these 2 genes, both HBA1 and HBA2, are present on which pair of chromosomes in humans? 16th pair of chromosomes. So a very important point. We can expect these kind of questions in NEET exam, because in previous exams it was not asked. So this year we can expect this question. So alpha thalassemia is caused due to 2 genes. And what are the names of those 2 genes? HBA1 and HBA2. And HBA1 and HBA2 are present on which pair of chromosomes? That is 16th pair of chromosomes. And why alpha thalassemia is caused? You know, 2 genes means 4 alleles, right? So alpha thalassemia is because of mutation in alleles of the gene, or directly deletion of the alleles of these 2 genes. So mutations on alleles, or by removal of one or more or deletion of one or more alleles of these 2 genes may result in the formation of alpha thalassemia. So what does it mean? Number of alpha chains produced itself gets decreased. And finally coming to beta thalassemia. In beta thalassemia, problem is always with only 1 gene. So beta thalassemia is associated with the problem of a single gene. And what do you call that gene? And that gene is called HBB. And what is the location of HBB? HBB is always present on 11th pair of chromosomes. Okay. So beta thalassemia is caused due to the defect in the single gene, and that gene is called HBB. HBB is present on 11th pair of chromosomes. So it is very clear, only 1 gene means 2 alleles, right? So what is the reason for beta thalassemia? Mutation in alleles of HBB gene. So mutation in alleles of HBB genes causes beta thalassemia. But for alpha thalassemia, if you ask me, mutation in the alleles of gene or deletion of alleles of the 2 genes may result in formation of alpha thalassemia. So in this way, thalassemia can be of 2 types, that is alpha thalassemia and beta thalassemia. In what pattern it inherits from parents to their children? Once again, in autosomal recessive pattern. Thalassemia is a quantitative problem, and what is the main thing happens in thalassemia? Very few globin molecules are synthesized. When number of globin molecules synthesized is decreased, blood hemoglobin level itself decreases and person develops symptoms of anemia. This problem is called thalassemia. And just for your information, remember, Once again we can classify thalassemia into, because alpha thalassemia is said, 2 alpha chains are there. So in medicine, doctors will classify thalassemia into alpha thalassemia minor, alpha thalassemia major. Similarly, beta thalassemia minor, beta thalassemia major, like that. And you need not remember all those at PUC level. Even for NEET exam, just remember one word. Remember this word, Cooley's anemia. What is Cooley's anemia? Beta thalassemia major. So all together, according to medicine, there are 4 forms of thalassemia. Out of 4 forms of thalassemia, the most severe form of thalassemia is beta thalassemia major. Condition of beta thalassemia major was reported by Dr. Cooley. So named after Cooley, beta thalassemia major is otherwise called Cooley's anemia. So out of all the forms of thalassemia, the most severe form of thalassemia is beta thalassemia major, which is otherwise called Cooley's anemia. So this is all about the 3 Mendelian disorders we have discussed in this video. What are the 3 Mendelian disorders I have discussed? Phenylketonuria, sickle cell anemia and thalassemia. And in my previous video, I have completed with hemophilia and color blindness. So with this, we have completed the topic gene disorders, which are otherwise called Mendelian disorders. Thank you.